Continuation of human evolution topic. In this video, we are going to learn about topics related to human evolution like bipedalism and brain size hypothesis about early human evolution. Before evaluating the changes that initiated the human lineage assigned to the genus Homo, it is helpful to outline the various hypotheses concerning relationships between the role of bipedalism and increase in brain size. We then will discuss the other major development, the origin of tool making and tool use. Charles Darwin said virtually nothing about human evolution and the origin of species. However, his second major book of evolution, The Descent of Man, published in 1871, dealt specifically with humans and their evolutionary origins. Ever since bipedalism, increase in the size of brain and the use of tools and language, which in combination with hallmarks of human evolution have been regarded as a set of related changes, each reinforcing the others. The fossil record, however, informs us that bipedalism arose much earlier than did the other two traits. However, at issue for some time has been whether the evolution of bipedal locomotion was an evolutionary innovation that facilitated changes in brain size and tool use. We can reconstruct enough of their environment to know that early hominins lived in woodland, dry grassland, and bush, where food supplies would have been seasonal. This, coupled with the patchy nature of the environment, would have selected for hominins who could adapt their diet to a wide range of foods, omnivores, and search for food over long distances. Evidence in the fossil record supports the hypothesis that standing upright and ability to move rapidly enhanced this lifestyle. The combined advantage of an upright stance and faster locomotion on the ground while retaining the ability to climb would have improved avoidance of predators. So, bipedalism would have fostered stick wielding and stone throwing actions enabled by the freeing of the forelimb from the duties of locomotion. Knowledge that chimpanzees use sticks as tools to obtain food has been used to develop a scenario that tool making in Homo, perhaps also in Australopithecus, began with using unmodified sticks and stones, from which the ability to modify those sticks and stones into more sophisticated tools arose. Next, origins of Homo. The genus Homo, which first appears in the fossil records in East Africa around 2.5 MIA, was similar in size to the Australopithecines but had a larger brain, 600 to 700 cm cube, and smaller molar teeth. The prevailing trend in the evolution of Homo, as an mammalian evolution as a whole, is of parallel lineages arising contemporaneously or almost contemporary related contemporaneously. Contemporaneously. I'm sorry. So, at least six species of Homo illustrate adaptive radiation in response to natural selection that took place in Africa over less than a million years. And Homo rudolfensis and Homo habilis. Even though the numbers of known specimens is small, they reveal that the skeletal change occurred rapidly in the Homo lineage. Many experts assign early East Africa Homo fossils to two species that occupied similar geographical regions, like Homo rudolfensis as early as between 2.4 and 1.8 MYA and Homo habilis between 2.0 to 1.6 MYA. The anatomical structure of the hands and feet of the early Australopithecines and early Homo habilis are consistent with the ability to climb trees. Homo rudolfensis and Homo habilis are the most likely members of the lineage that give rise to the next two species of Homo, which appear in the fossil record 1.9 MYA, that is, no more than 500,000 years, as little as 1,100 years after Homo habilis and Homo rudolfensis. One of the species lived in Africa and the other in Asia. Both were taller and had larger brains than their preceders. The species are Homo ergaster and Homo erectus. Then Homo ergaster and Homo erectus. The African species Homo ergaster was taller and leaner with a brain capacity of 7,050 to 1,100 centimeter cube and a large body size than Homo habilis and Homo rudolfensis, either of which could have been its ancestor. So later, Homo ergaster used fire and large hand axes of Herculean type. Whether these attributes indicate group living and hunting is hard to determine. As with Lucy, the almost complete skeleton of this species, Narioctome skeleton is the most complete fossil of a juvenile male, which is represented with the help of this picture. A second species, Homo erectus, contemporaneous in time with Homo ergaster, was first discovered in Java. A thicker skull and heavier brow ridges, along with the different geographical distributions, allow the two species to be differentiated. Brain volumes of the two species overlapped, and both species are associated with Herculean tools. Later discoveries showed that Homo erectus was present over a wide geographical area in Africa, China, and Europe, and persisted until as late as 50,000 years ago in Java. Fossils discovered in 2004 from the islands of Flores in Indonesia reveal what has been interpreted either as a related population of dwarf Homo erectus or as a new species Homo floresiensis that existed as recently as 18,000 years ago. Such a recent existence of human species other than our own is a remarkable suggestion that highly controversial. Some experts claim that the best preserved individual is more modern, a dwarf human, Homo sapiens, but with signs of a congenital malformation, a topic taken up later. So, Homo sapiens and Homo heidelbergensis. One of the reasons for Homo erectus receiving so much attention and a partial explanation for the controversy over the Flores individuals, that is, our species, Homo sapiens, has been traced back to a lineage that contained Homo erectus. Variations among specimens of Homo erectus and the often but subtle differences between Homo erectus and Homo sapiens led some experts to claim that Homo erectus or some variants within Homo erectus are the best manifestations of Homo sapiens. Part of the variation within Homo erectus reflects individuals in different geographical locations that would have existed as distinct and potentially non interbreeding populations. Some conclude that the variation can in part be solved by recognizing another species, Homo heidelbergensis, from fossils in England and Germany, dated to about 200,000 years ago. The specimens have larger brain volumes than and other features intermediate between Homo erectus and Homo sapiens. To complicate things further, this Homo heidelbergensis may have arisen from a population of Homo ergaster and not from the more geographically widespread Homo erectus, which persisted in parts of the world long after Homo heidelbergensis arose. So the tree of Homo evolution, therefore, is much more like a shrub with many branches than it is like a tree with a single trunk. Instead of a ladder with humans at the pinnacle, there is a bush with humans as one little twig. As you will see as you read on, the tree is about to become even bushier. Why? For two reasons. Another species, Homo neanderthalensis, discovered in 1856 in the Neander Valley, Germany, arose from European populations of Homo heidelbergensis. Many paleontologists have concluded that Homo sapiens evolved from Homo heidelbergensis, separation of the two lines having occurred in Africa around 200,000 years ago. Then Homo sapiens, or humans. Many older texts uses terms such as primitive and advanced or higher and lower when referring to earlier and later forms within a lineage. Such terms are no longer regarded as appropriate, conveying as they do the notion of evolutionary progress on some subjective scale. The term anatomically modern humans is now used for extant and fossil individuals of the species Homo sapiens. Anatomically modern humans. It's inappropriate to say primitive, advanced, higher, lower forms. We should say anatomically modern humans for Homo sapiens. The earliest human evidence of Homo sapiens is located in Africa, a finding reflected in the out of Africa hypothesis for the spread of Homo sapiens around the world, as discussed later. Then, three individual specimens of anatomically modern human discovered in 1,60,000 years old deposits at Herto, Ethiopia are the geologically oldest members of Homo sapiens. More recent fossils from Mount Carmel in Israel date to 90,000 years ago. Mauritian stone tools are associated with both sites. In even more recent deposits, 35,000 years ago during the Upper Paleolithic, Paleolithic, Paleolithic in Europe, the features of human fossils and the tools present both had changed. Pro ridges decreased, skull vaults were higher, reflecting changes in brain size and form. Faces were smaller and less protruding, and flanked flint tools of the Orignation era were present. Humans were now representing their environment, especially the animals in it, as paintings on cave walls and as sculptures in bone. Next topic, Neanderthalensis, another species of Homo with comment on Denisovans. Neanderthalans, Homo neanderthalensis, were widespread in Europe and Western Asia from 30,000 to 300,000 to 30,000 years ago. Shorter than modern humans and with brains that were 10% larger, Neanderthalensis had distinctive broad ridges, large jaws, small chins, robust skeletons, and other anatomically important features not seen in humans. So, like humans, Neanderthalans used stone tools to hunt large animals, bears, and mammoths, as shown in most museum reconstructions and dioramas. From flowers found on the graves of dead individuals, it has been concluded that Neanderthalans performed rituals, and 78 bones from 6 individuals who lived 1000 to 1200 years ago provide evidence that Neanderthals practiced cannibalism. The bones scattered around the hearths of 3 fires show cut marks and evidence that the bones had been torn apart, both of which are consistent with dismembering because the Mauritian culture is associated with Neanderthals and with Homo sapiens. Some have concluded that there may have been cultural overlap between the two species. Parallel tool uses does not necessarily mean cultural overlap, however. To add further complication, in 2010, a previously unknown population of Arctic humans were described to have inhabited the Devizona cave in Siberia 30,000 to 50,000 years ago. The cave also contains Neanderthal fossils and Mauritian tools typical of those made and used by Neanderthals dated to 45,000 years ago. The cave was later occupied by humans also. Genomic sequencing of the Devisonians, as they are known, confirmed them as Arctic human separate from Neanderthals, but also showed that some 5% of the DNA in living Australian Aboriginals and Malaysians are derived from Denisovans. Then Neanderthalans, another species of Homo with comment on Denisovans, like species status. The difficulty of assigning species status was discussed in the text in relation to Homo erectus, and the Flores specimen was also elaborated further and later. Similar difficulties are encountered with Neanderthalans. Some physical Anthropologists have concluded that skeletal evidence, thicker skulls, differences in the pelvic girdle, and the presumed limited language ability of Neanderthalensis support Neanderthals as a subspecies of Homo sapiens designated Homo sapiens neanderthalensis, although Neanderthal faces and their large deep rooted teeth do not align them with any human populations. The Neanderthal genomes. In recent years, sufficient Neanderthal DNA has been isolated and sequenced. When analyzed with sophisticated methods to estimate missing DNA, a reasonable first approximation of the Neanderthal genome was constructed. One study used 1 million base pairs of Neanderthal human have around 3.2 billion base pairs of DNA. The other 65,000 base pairs. These studies have been described as perhaps the most significant contributions since the discovery of Neanderthalensis 115 years ago. The 1 million base pair study produced an estimate of divergence of humans from Neanderthalensis many years ago, later replaced by and not integrated with Homo sapiens. Homo sapiens began to replace Neanderthals in various parts of the world around 40,000 years ago. Neanderthals are estimated to have died out 28,000 to 24,000 years ago. How do we know that Neanderthals died out rather than integrating with Homo sapiens? The two species certainly coexisted between 500,000 years and 24,000 ago, and both overlapped with Homo erectus between 500,000 years ago and many years ago. Indeed, mixing of artifacts in the reindeer cave in France means that assignment of ornament to, or rituals to Neanderthals has to be done with caution. Such artifacts may have been generated by Homo sapiens. Strong molecular evidence against integration comes from the recovery and analysis of Neanderthal mtDNA sequences, which show little evidence of any contribution of Neanderthal of human mtDNA. mtDNA or mitochondrial DNA isolated from a 38,000-year-old Neanderthal skeletal is significantly more different from human mtDNA than expected if it were a sample of normal human variation. According to the data from the study, Neanderthal and human mtDNA lineages diverged from 660,000 to 140,000 years ago, and Neanderthals went extinct without contributing mtDNA to modern humans. Then developing Neanderthals. Information from CT scans or computed tomography on Neanderthal pre- and postnatal development has been obtained: a neonate from a cave in Russia and two infants from a cave in Syria, and compared with developmental stages of anatomically modern humans. Both species share similarly large brain sizes at birth, with Neanderthal brains growing faster during infancy than human brains, and Neanderthal brain size increasing rapidly in the first few years after birth. An even more recent study using a method that allows very precise aging shows that tooth development was faster in Neanderthals than in humans. It seems reasonable to conclude that Neanderthal fetuses developed and matured faster than human fetus, although both species show lengthening of parental development when compared with other hominins. Although the weight of evidence supports Neanderthals and humans are separate species, we await even more detailed studies of those and other organ systems. Later, tool used by species of Homo. Individuals in the genus Homo developed the capacity to make and use stone tools, the oldest of which are dated to 2.5 MYE. Tool making and use has been classified into a series of 4 stages, discussed with the help of some other additional information which we will be sharing. Then each state is associated with a particular species of Homo, beginning 2.5 MYE with Homo habilis and extending to 200,000 years ago with Homo sapiens. These tools were used to manipulate plant material and carve up small reptiles, rodents, pigs, and antelopes, and scavenge the remains of larger mammals. And then out of Africa theory. There are two main views of origin and subsequent dispersal of modern humans from Africa. The single origin hypothesis, also called out of Africa or Noah's Ark model, proposes the origin of Homo sapiens in a single geographical region, Africa, followed by dispersal to other continents. Then the multiple origin hypothesis, also called candelabra model, based on its shape, proposes parallel origin of Homo sapiens in different unconnected localities. So, accurate determination of the date of the last common ancestor of modern humans is a crucial piece of evidence in deciding between the two hypotheses. Existence of the most immediate ancestor, one or more MIA, would coincide with one of the dispersals of Homo erectus from Africa. It would indicate that modern humans found in different continents represent evolutionary lineages that each began from Homo erectus in geographically separated localities and so support the multiple origin hypothesis. If the most immediate Homo sapiens ancestor was much more recent, say 100,000 to 500,000 year old, then the dispersal of Homo sapiens would have occurred in area occupied by Homo erectus after 1 to 2 old MI-old dispersal of Homo erectus and so support the single origin hypothesis. So this picture shows diagrammatic representation of two models of the origin of Homo sapiens. Like first, in case of human, blue color, originated in one locality and migrated to other continents where they replaced Homo erectus populations, brown color, that had entered these continents. Later, humans, blue color, originated in blue color representation, originated in different localities independently of their species, other species, like out of Africa model, like single origin of Homo sapiens in Africa and replacement of Homo erectus in Europe, Africa, and Asia. Then candelabra model. Multiple origins of Homo sapiens. I am not sure about the pronunciation. Sapiens from Homo erectus populations in Europe, Africa, and Asia. Next topic, mitochondrial Eve. An important data set helping to resolve the immediate ancestor of Homo sapiens is mitochondrial DNA from a large number of extant humans representing many populations. Two major findings are consistent with a single origin hypothesis. All of the non-African mtDNA sequences are variants of the African sequence, and most of the variability in mtDNA sequences occur among members of African populations. Both such findings are consistent with the oldest mtDNA sequences being in African populations. Multiple populations evolving in parallel would be expected to show similar amounts of sequence variability. So in 1987, on the basis of mtDNA nucleotide sequence analysis of geographically diverse human populations, Rebecca Cann and co-workers in 1987 proposed a 200,000-year-old common mitochondrial DNA ancestor, mitochondrial Eve, of modern humans, a finding consistent with the single origin hypothesis. Subsequent studies, in which calibration of age of human origination was based on similar mtDNA sequence data, provided further support for the single origin hypothesis, although molecular clocks are difficult to calibrate. One major conclusion from this analysis is that all modern human mtDNA sequence originates from a single ancestral sequence in Africa between 1,40,000 and 2,90,000 years ago. Each geographical group outside Africa includes more than one mtDNA branch, that is, each arose from more than one population, each with distinctive mtDNA. 9 major descendant sequence were distributed to other geographical regions through migration. Further sequence changes occurred over time in populations in those regions. Highland people of New Guinea have 7 maternal origins, meaning that individual geographical regions were colonized more than once. As the rate of mtDNA sequence divergence can be estimated 2 to 4 point nucleotide change in vertebrates, though slower in though slower in primates. Timing of waves of colonization also can be projected with the help of this figure. This shows phylogeny of mitochondrial DNA from 147 humans from 5 geographic regions. The ancestral sequence is designated as each node in the phylogeny indicates a major descendant sequence. Estimated dates for each node thousands of years ago are given in parentheses. Areas colonized by each sequence is indicated in the top of the figure. So even though such estimates are influenced by the ways in which fossil species are arranged phylogenetically, the hypothesis of a single African origin of Homo sapiens remains the best explanation for the origin of anatomically modern humans. Then Y chromosome Adam. Empty DNA sequence are not complicated by the recombination of genes from the male and female parent. Empty DNA sequence track the female genome back in time. Interestingly, perhaps even surprisingly, Y chromosome, which is inherited to the male line, also contains phylogenetic information, but of a different nature to that seen in empty DNA. So, ordinarily, sequence changes in nuclear genes would be hard to differentiate into those resulting from recombination and so potentially recent, and those that are much older. So, fortunately, a large proportion of the human Y chromosome does not participate in recombination. As a consequence, mutations build up in regions over time. The phylogenetic analysis of more than 1,500 individual humans from all continents traced Y chromosomes to a common African ancestor living and in Africa. So, both mitochondrial Eve and Y chromosome Adam confirmed as African origins of humans. Even the margins of errors in such estimates 1,50,000 for Y chromosome Adam and 1,40,000 years for mitochondrial Eve are remarkably close. So this figure, one scenario for the geographic distribution of Homo sapiens from their African origin. The numbers derived from the study of genetic distances between 26 human populations and represent estimated dates thousands of years ago when this populations reached their various destinations. The migrations correlate highly with many major patterns of linguistic evolution. Then autosomal human. The phylogenetic analysis by Ney and Roy Chowdhury in 1993 of 29 autosomal genes, nuclear genes not located on the sex chromosomes from 26 populations support a single African origin of humans around 2 lakh years ago. According to this data, dispersal resulted in humans reaching Eastern Europe 50,000 to 70,000 years ago, Australia 40,000 years ago, North America 12,000 years ago to 14,000 years ago, in South Africa 13,000 years ago. Humans did not undergo such vast migration without adaptation to and interactions with their environment, developing social structures and enhancing their ability to communicate using speech and language. Then humans as hunter-gatherers. Next topic. Humans as hunters or gatherers. Hunting in groups or packs did not evolve with humans. Jackals and hyena hunt in packs as their major means for capturing prey. Chimpanzees and baboons also occasionally form groups to hunt smaller animals. Grouping of 2 to 5 male chimpanzees will tree a monkey and cut off its escape by positioning themselves around it. So the earliest member of Homo sapiens moved from forest to savanna as they roamed over large areas in search of food and prey items that were patchily distributed spatially and seasonally. Exposure to differing environments during migration to the food types in those environments and the discovery of fire and cooking would have selected for structural and behavioral changes and adaptations. For example, anatomical and inferred physiological evidence has been used by Bramble and Lieberman in 2004 to conclude that adaptations for long distance endurance running separate Homo sapiens from Homo erectus. Analysis by Rowland et al. In 2010 for covariation and correlations of toe and finger lengths in 89 extant individual common chimpanzee and two not two humans. Supports coevolution of human but not individual common chimpanzee hands and feet. Elongation of human toes facilitated bipedalism and running. Elongation of human fingers facilitated changes in the manufacture of tools. Co-occurrences of human and other animal bones in East African fossil sites are interpreted to, interpreted to mean that scavenging was established 2 MYE. This early date is interpreted as indicating that humans were scavenging kills made by other large predators. Evidence indicates that hunting and gathering arose in humans much more recently than scavenging, on the order of 55,000 years ago. A single kill of a large mammal would have provided food for more than one individual or more than a day if kept away from other scavenging mammals. Such kills would have been passed on to the next generation by imitation, and later as social living becomes more established by active teaching or hunting skills and of locations of seasonal populations of prey. A hunter gatherer society had begun. So that's all about this topic. Thank you for listening.